Welcome back to the Deep Dive. Today we're getting into something pretty amazing, a world that's usually invisible but has a huge impact. Fungi.
Absolutely. Think about, the mold on that forgotten orange in your fridge or the blue veins in Roquefort cheese.
Or even something less pleasant like athlete's foot. Fungi are just everywhere. They shape our food, give us vital medicines, but sometimes cause serious problems too. It makes you wonder, how do these apparently simple things do so much?
It's fascinating, isn't it? They're constantly working, often unseen, interacting with everything around us. Silent architects, sometimes silent threats.
Totally. And for this deep dive, we're zeroing in on a massive, super diverse group, the filamentous ascomycetes. We're drawing heavily from a foundational chapter in an introductory mycology text to pull out the key stuff for you.
Yeah, our goal today is to really unpack their basic biology. We're talking structure, reproduction, genetics, how they're classified, and maybe most importantly, why they matter, in the environment, for our health, even in what we eat.
We'll break down the complex bits, make it digestible, so you really get what's important about these microscopic powerhouses.
Exactly.
Okay, let's unpack this. So when we talk fungi, the phylum Ascomycota is like huge, one of the biggest groups. And within that, we're focusing on these filamentous ones. They tend to look more complex structurally than yeasts, right?
That's right. They're not just single cells, they form threads. And a key thing about them is how they handle sexual reproduction. Many develop distinct sex organs, though some are kind of losing those bits over time.
Sex organs in fungi.
Well, functional equivalents. Gametangia. There's often a female structure, the ascogonium, and then structures that arise from it, like ascogenous hyphae, and these cool little hook-like things called croziers. These lead to the formation of the actual spore sacs, the asci.
Okay, so these asci are where the sexual spores are made. Or are they just out in the open?
Usually not. They're typically tucked inside a protective structure, a fruiting body called an ascocarp. Think of it like a little container.
And these containers come in different shapes.
Yeah, several main types. There's the Cleistothecium, which is totally closed, like a little ball. Then the Parathecium, more flask-shaped with a little pore at the top for spores to get out. An Apothecium is open, cup-shaped, maybe like a tiny, tiny mushroom cap. And sometimes the asci are embedded in a sort of fungal tissue mass called an ascostroma.
So the shape relates to how they release spores.
Pretty much. Strategy. Though in a few cases, there's barely an ascocarp at all, just a loose tuft of hyphae.
Okay, that's the sexual side. What about just growing and spreading fast?
For that, they rely heavily on asexual reproduction. They pump out huge numbers of asexual spores, different kinds, generally called diaspores or conidia. Makes them spread like wildfire when the conditions are good.
And genetically, are they all related?
That's a key point. Ribosomal DNA sequencing, looking at their genetic code, shows that these filamentous ascomycetes form a monophyletic group. Meaning they all evolved from a single common ancestor. They're a genuine evolutionary lineage, separate from other fungi like yeasts or the really early diverging groups.
Okay, so we have these complex structures, different ways to reproduce. How does this all fit together in their sort of day-to-day existence? What's their life cycle actually look like?
Right, so the typical pattern starts with the sexual spores, the ascospores. They get released, find a good spot, and if conditions are right, they germinate. Each one sends out a little thread, a germ tube, which grows and branches to form the main body, the mycelium.
A network of threads.
Exactly, and that mycelium is the workhorse. It starts producing structures like conidiophores, which churn out those asexual spores, the conidia, in massive numbers.
That's the rapid spread part.
That's the rapid spread part. They can go through that asexual cycle over and over during a growing season, but then maybe conditions change, temperature, moisture, nutrients, and that triggers the sexual phase.
Right, back to the ascocarp.
Back to the ascocarps. The mycelium starts forming them, the asceliums develop inside, produces ascospores, and the cycle is ready to start again. And for getting through tough times, like winter, they often survive as dormant mycelium or maybe ascospores, waiting for things to get better.
It's a really adaptable strategy, survival and spread. Now, given all this diversity in structure and lifestyle, how on earth do scientists classify them? I bet that's been tricky.
Oh, absolutely. It's evolved a lot. Historically, mycologists had to rely on what they could see under the microscope, the shape of the ascocarp, how the asci release spores, the presence of sterile threads called paraphyses, even what the fungus was growing on.
Stuff you could observe directly.
Exactly. And some of those features are still useful, but our understanding has gotten much deeper.
So there were attempts to build systems based on this morphology.
Yes, definitely. Pioneers like Nanfelt and Luttrell back in the mid-20th century developed influential systems. They grouped fungi based on how the ascocarp developed and its final structure.
Like those different shapes you mentioned?
Right. So fungi with those closed Cleistothecia often fell into a group called plectoscales. We still sometimes informally call them plectomycetes. Those with perithecia or apothecia were grouped into things like ascohymeniales, and those with ascostromata were ascoloculares. These formal names are less common now, but the concepts, like plectomycetes, stick around because they're convenient descriptors.
Okay, so that was based on visible structure. But you said our understanding deepened. What changed?
This is where it gets really interesting. Molecular data. DNA sequencing, basically. It revolutionized taxonomy.
How so?
Well, sequence analysis lets us look directly at evolutionary relationships.
Hmm.
And it turns out sometimes fungi that look similar, maybe because they have the same type of ascocarp, aren't actually that closely related, and vice versa. It completely reshuffled the fungal family tree.
So looks can be deceiving.
Totally. It blew up old ideas about which fungi were primitive and which were advanced just based on structure. The DNA tells a much more complex story.
So for our discussion today, how are we approaching classification?
We'll use a mix. We'll refer to some of those informal groups, like plectomycetes, because they're helpful categories based on shared features discussed in our source text. But we always have to keep in mind that the molecular picture is the ultimate guide and is constantly being refined. And today, we're definitely putting a spotlight on those plectomycetes.
Great. Let's drill down into the plectomycetes then. What really defines them and what are some key examples?
Okay, plectomycetes. Generally, the spore sacs are thin-walled, often sort of globose or pear-shaped, and critically, they're often evanescent.
Meaning they just dissolve.
Yeah, they basically break down after the spores inside are mature, releasing the spores within the ascocarp. And the assae themselves are usually scattered around inside, not lined up neatly in a layer like you see in some other groups. The ascospores are typically simple, just single cells.
And the defining feature is that closed ascocarp, the Cleistothecium.
Often, yes. When they make an ascocarp, it's typically a Cleistothecium completely enclosed. But the wall of that Cleistothecium, the peridium, can be incredibly varied. Sometimes it's just a loose web of hyphae, other times it's a really solid, complex structure, almost like a tiny cage.
And they have asexual forms too.
Oh, yes. Various types, often very characteristic asexual spores or conidia. And importantly, while that closed ascocarp made people think they were maybe primitive.
You said the DNA refutes that.
Exactly. Modern evidence shows that's not the case at all. They're a successful, diverse group, just with a different strategy.
And hugely important, you mentioned.
Immensely, economically, ecologically. This group includes animal pathogens, vital antibiotic producers like penicillium, but also fungi that make dangerous mycotoxins, and key players in food fermentation. A real mixed bag.
Okay, let's look at an order within them. Ascospores. Sounds obscure.
It might seem so. They're interesting because they sometimes get mistaken for yeasts, partly because they don't have a typical elaborate ascocarp, but the DNA evidence puts them firmly in the plectomycetes.
And they have a connection to bees.
A big one. The genus Ascosphaera is famous, or infamous, for Ascosphaera apis, the cause of chalk-brood disease in honeybee larvae. It's a major problem for beekeepers.
Wow. And they're tough little fungi.
Incredibly so. They're known for tolerating or even needing environments with very low available water. Think super sugary or salty conditions. Growing them in the lab might require like 30–40% glucose.
That's intense.
It is, and their spores are packaged uniquely too, grouped into spore balls inside a clear spore cyst. Reproduction is also interesting. Apis is dioecious.
Needs two different individuals.
Right. A special receptive hypha from one has to fuse with a hypha from a compatible male type. Then the sexual structure develops. But other species, like Aeatra, are homothallic self-fertile.
Okay, and what about Ermascus, another one mistaken for yeast?
Yes, also lacking a proper ascocarp. But even early on, structural details like warren in bodies and their hyphal walls hinted they weren't yeasts. Ermascus fertilis has lots of hyphae and can make these unusual thick-walled asexual spores by rexolytic secession, basically. The cell wall splits.
And sexually.
It's simpler. Two compatible branches just fuse, nuclei fuse, and the resulting zygote develops directly into an ascus with 8 spores. No complex fruiting body.
All right, let's shift gears to another really significant order, the Anagenales. You said this one has major medical importance. and some weird abilities.
Yes, the Onygenales are a big one, medically speaking, and their standout feature is that many species can degrade keratin.
The stuff in hair and nails.
Exactly, that tough protein. This ability is key to their lifestyle, explaining why you find them on animals, or associated with things like dung, feathers, skin flakes, even bone. Structurally, their asci are often spherical and deliquescent. They dissolve to release spores. The spores themselves are single-celled but can be very ornate, and their asexual conidia often show that same unusual splitting mechanism, rexolytic succession.
So that keratin-degrading ability must link them to diseases, right?
Directly. Within the family Onygenaceae, we find some major human pathogens. Take the genus Agilomyces. A. dermatitis and A. capsulatus are classic examples. They are dimorphic.
Meaning they change shape.
Right. They grow as yeast-like cells at human body temperature inside a host, but as filamentous mold, mycelia, at cooler temperatures, like in the soil, a two-faced existence.
Clever adaptation. What diseases do they cause?
A. dermatitis causes blastomycosis. It can affect the skin, but usually it's a lung infection from inhaling spores, sometimes looking like TB. It can spread and be fatal. It's endemic in parts of North America, also Africa, and dogs get it too. Often found in moist soil near rivers.
And E. capsulatus.
Causes histoplasmosis, very widespread. Again, typically a lung infection, but can disseminate, especially if your immune system is weak. It thrives in soil enriched with bird or bat droppings. Think caves, old chicken coops. Spelunking can be a risk factor.
Wow. Then there's Cochliobolus. That one sounds familiar.
Yes, valley fever, another really important pathogen, especially in arid regions like the U.S. Southwest. We actually don't know its sexual stage. It produces these asexual spores called arthroconidia in the soil. You inhale them, they swell up in the lungs into structures called spherules, which then release more infectious cells.
Sounds nasty.
It causes a flu-like illness. Most recover, but it can be severe, especially in certain populations. And a big warning. It's highly infectious in the lab because it makes clouds of those tiny arthroconidia. Needs careful handling.
Definitely noted. Are there other families in Onygenales linked to disease?
Yes, the Arthrodermataceae. Also keratin degraders, but they have smoother spores and these unique kind of bone-shaped swollen cells in their cleistothecium walls. This family is home to the dermatophytes.
The ringworm and athlete's foot culprits.
Exactly. The fungi causing dermatophytoses, those common skin, hair, and nail infections. We often classify them based on where they primarily live. Geophilic are soil fungi that sometimes jump to humans. Zoophilic are mainly on animals but can infect us. And anthropophilic are pretty much restricted to humans.
Is there an evolutionary trend there?
There's a hypothesis, yeah, that they evolved from free-living soil saprobes, adapted to animals, then specialized on humans, often losing sexual reproduction and becoming more host-specific along the way. Think of trichophyton rubrum, a super common cause of athlete's foot passed around in locker rooms. That's anthropophilic. Or Microsporum canis, causing ringworm you can get from your cat or dog. That's zoophilic.
Okay, let's switch tracks now to the Uroshalus. You hinted we probably encounter these ones a lot.
Oh, definitely. Think common molds, maybe even medicines you've taken. Uroshells typically have those spherical, dissolving asci. Their asexual spores are usually dry and separate cleanly, often produced from vase-shaped cells called phyalides. They love growing on starchy, oily, or cellulosic stuff, basically, lots of food sources.
And within this order, the family Trichocomaceae sounds like a real heavyweight.
It absolutely is. This family is incredibly common, found everywhere. They're often called fungal weeds because they grow fast, produce tons of spores, and colonize things quickly. And they have this amazing dual nature.
Friends and foes.
Exactly. On the friend side, think food. Aspergillus and penicillium are crucial in making Asian fermented foods like soy sauce, miso, sake. Monascus species ferment rice to make red food coloring.
And cheese.
And cheese. Penicillium roqueforti for Roquefort. P. camembert for Camembert. They give those cheeses their characteristic flavors and textures. They're even involved in curing some meats. Industrially, they're powerhouses too. Aspergillus niger makes huge amounts of citric acid for soft drinks. They produce all sorts of enzymes used in detergents, food processing, you name it.
And the big one, antibiotics.
The game changer. Fleming's discovery of penicillin from a penicillium mold contamination is legendary. Penicillium chrysogenum is the main industrial producer. And there's that story, maybe partly legend, about Moldy Mary finding a super productive strain on a cantaloupe in Peoria during WWII. Another key antibiotic, grisafulvin for fungal skin infections, also comes from a penicillium.
Amazing benefits. But you said foes, too. the mycotoxins.
Yes, the dark side. Aflatoxin, from Aspergillus flavus primarily, is notorious. It caused a huge turkey die-off in the UK in 1960, the turkey X disease. Aflatoxin B1 is a potent liver carcinogen. There's even speculation it worsened mortality during historical plague epidemics by weakening people's immune systems.
It's chilling. Are there others?
Plenty. Ochratoxins, tremorogenic toxins, citrinin, patulin. They can damage kidneys, cause neurological issues. It's important to remember these toxins likely evolved as defenses against insects or other microbes trying to eat the fungus or compete with it.
And they can directly cause infections in humans, too.
Yes. Aspergillus species like A. fumigatus, A. flavus, A. niger cause aspergilluses. This can range from allergic reactions to fungus balls growing in lung cavities or sinuses to devastating invasive infections where the fungus actually grows into tissues. This is especially dangerous for immunocompromised people and can be fatal. A. fumigatus loves compost heaps, by the way.
What about penicillium species as pathogens?
Less common than Aspergillus, but Penicillium marneae is a significant one, especially in Southeast Asia. It's another dimorphic species, unusually yeast-like in the body for a penicillium. It causes penicilliosis, which can spread through the body and be fatal, though it is treatable if caught early.
So lifesavers and killers. And just plain annoying spoilers, too, right?
Oh yeah, huge spoilage agents. Penicillium italicum, blue mold, and P. digitatum, green mold, on citrus fruits are classic examples. P. expansum rots apples. They ruin stored grains, silage for animal feed, leather goods, fabrics. Ever heard of mildew? That's often them. Controlling moisture is crucial to keep them in check.
Let's look closer at their asexual reproduction, especially Aspergillus and Penicillium. How do they make all those spores?
Okay, so their main body, the mycelium, is this network of branched septate cross-walled hyphae. Sometimes they form sclerotia, these hard, dormant masses that help them survive tough times. For Aspergillus, the spore-producing structure, the conidiophore, is quite distinctive. It arises from a special foot cell in the mycelium, grows into an upright stalk, and swells at the top into a bulbous structure called a vesicle.
And the spores come off that vesicle.
Directly. In some species, that's called uniseriate. In others, biseriate, there's an extra layer of cells called medullae on the vesicle, and then the spore-producing phyllades arise from those. Either way, the phyllades produce spores, conidia, and long chains, with the youngest spore at the base pushing the older ones out like beads on a string. Interestingly, trace elements like copper can dramatically affect the spore color in species like A. niger.
And Penicillium, you said it looks different.
Yeah, Penicillium has these characteristic branched conidiophores that look like little brushes or brooms, hence the name penicillus, Latin for small brush. The branching patterns are really important for identifying species, but in both genera, the conidia themselves are usually globose or ovoid and give the colonies their typical green, blue, yellow, sometimes other colors.
Okay, that's asexual. What about the sexual side in this family, Cochliobolus?
Yeah.
Is it as varied?
It is quite varied, yes. A common pattern seen in the genus Eurotium, which is the sexual stage of some Aspergillus species, involves the male and female structures coiling together. Nuclei pair up, specialized ascogenous hyphae grow out, and these produce the asci scattered within the developing Cleistothecium. And again, those asci are typically evanescent, they dissolve, leaving the spores loose inside.
And other.
Monascus, the red rice fungus, is similar, but the wall around the asci is very thin. In Emericella, another Aspergillus sexual stage, the Cleistothecia are often embedded in a denser fungal tissue, a stroma, and covered by these unique, thick-walled, swollen cells called Houle cells, which can actually function as dispersal units themselves. Some Emericella have beautifully ornamented red ascospores.
What about Eupenicillium, the sexual stage for penicillium?
Right. In Eupenicillium and the related Hemicarpentellus, the asci develop directly within a hard stroma-like structure rather than having a distinct Cleistothecium wall inside the stroma. The asci might be single or in chains.
So lots of variation in the packaging. What about the spores themselves?
In many of these genera, Eurotium, Emericella, Eupenicillium, Monascus, the ascospores often have a distinct shape. They're oblate, meaning flattened and bivalvate, with a groove around the equator. They look like tiny pulley wheels and split in half when they germinate.
But not all Eurotiales are like that.
No. Genera like Talaromyces and Bicyclamis are different. Their asci are surrounded by just a loose network of hyphae, not a dense stroma or distinct Cleistothecium wall. And their ascospores are usually spherical or ellipsoidal and just swell and burst to germinate without that pulley wheel split.
Okay, one last quick stop. A family called Pseudothecia. What's their story?
Right, a less commonly discussed family, but distinct. They also often have coiled beginnings to their sexual structures, but their Cleistothecia tend to be clear, hyaline, or dark-colored, with walls made of multiple layers, unlike the often bright pigments in Trichochomaceae. Their ascospores are simple, single-celled, spherical, or ovoid, and lack germ pores.
And their lifestyle. Where do they hang out?
Often in different niches than the Trichochomaceae, you might find them on dead wood, dung, or maybe composting plant material. An example is Cryptondoxula hypofloia, found under bark, which has a cleistothecial wall made of distinct plates.
So wrapping this all up, what's the big takeaway for our listeners?
Well, we've really journeyed through this incredibly diverse group, the filamentous ascomycetes. We've seen their intricate structures, their complex life cycles, and their really surprising dual roles, crafting our food, giving us life-saving drugs, but also acting as pathogens and spoilers.
Yeah, and we saw how our understanding, our classification of them, has changed dramatically with new tools like DNA sequencing. It shows how even these seemingly simple molds have incredibly complex lives and profound impacts, both good and bad, on us and our world. They're a huge part of that hidden microscopic engine driving so much of life.
It really makes you think. Given how incredibly adaptable these fungi are, surviving in extreme conditions, breaking down tough materials like keratin, what other roles might they be playing that we haven't discovered yet, especially in a changing world? What secrets might still be locked in their genes for future medicines, biotechnologies, or even future challenges?
That's a great thought to leave folks with. We really hope this deep dive has given you a new appreciation for this hidden world in the fungi that truly shapes so much of our existence.
Thanks for joining us on this fungal exploration.
And thank you, as always, for being part of the Last Minute Lecture family. We'll catch you on the next deep dive.